This is the first Mission 7 experiment. This is a flight experiment, grade 12. So these are the experiments that have not yet flown and should have been in orbit right now, except that we lost SpaceX 7.
Hello everyone, my name is David Washington, and I am a co-principal investigator for the Verne, California, Damien High School SSEP group. And our experiment name is Tardigrade Cryptobiotic Strategies versus Microgravity. Now before we go on and actually talk about our presentation, we'd like to thank a few people who without them, our experiment would not even be possible. First, we'd like to thank some of our most generous benefactors, the Amundsen Foundation, Dr. James M. Lawley, Dr. William Saito, Dr. Tostianis, and Ms. Martina Ramirez. Next, we'd like to thank our teacher facilitator and our biggest supporter, Ms. Charity Trojanowski. Without her continued support to bring the SSEP group to Damien High School, we would all be aspiring scientists and seniors who would not really know what we'd be getting into going into college. But through her help and through SSEP, we are greatly more aware of what real science is in the real world, and we're eternally grateful for her for that. We'd also like to thank our science department head, Ms. Angela Curry, and our English teacher, Dr. Michael Williams, for helping us write our proposal. Last but certainly not least, we'd like to thank Professor Jonathan Wright from Pomona College for letting us use his lab and some of his materials, and even his research on tardigrades to help us perfect our procedure, and as well as ask him for any help along the way. Now onto our experiment. I'm joined by my colleagues, fellow principal investigator Garrett Souza, Ashwin Balaji, investigator Richard Conti, and collaborator Aditya Patel.
All right, so our experiment is predicated on the tardigrade, which is a microscopic aquatic organism. And what makes it special is its ability to essentially shut down all metabolic processes when it's encountering a volatile environment or an adverse condition. And even more so, it is special because it is able to replicate all 5 types of cryptobiosis, which is this shutting down of metabolic processes. So the 5 types of cryptobiosis are anhydrobiosis, in which you're able to shut down your body when there's a lack of water, and oxybiosis, you're able to shut down with a lack of oxygen. Chemobiosis, you're able to shut down if there are harmful solutes in the environment. Osmobiosis, when there are high levels of toxins, as well as cryobiosis, when there's low temperatures. Specifically, we studied anhydrobiosis. Now, these cryptobiotic abilities are incredibly uncommon on Earth simply because we don't really encounter this kind of volatile environment, so it would not be evolutionary necessary. Which leads several scientists to point to a possible extraterrestrial origin to the tardigrade. And that is an even more important reason to study them. Now, once they undergo this cryptobiotic ability, they enter into what is called a ton state, as can be seen in the picture here. They convert all sugars in their cells into what is called a trehalose sugar, which is a gel-like substance and is able to preserve the cell. And they also collapse all limbs of the body and enter into this pill-like or ton state. And once hospitable conditions are reintroduced, the process is reversed and they are able to resume functioning as if nothing ever happened. In our initial ground truth experiment, 20 of 23 specimens were able to enter into this cryptobiotic state and then resume activity after 10 days in cryptobiosis. So for our experiment, we had 2 volumes. We used the type 2 FME2, and one volume had desiccated or dehydrated tardigrades on wax paper. We artificially did it in a lab, and the other had naturally desiccated tardigrades on pieces of moss.
Our experimental procedure is set up so that way we have 2 sets of tardigrades on the International Space Station and 2 sets here on Earth. Each set will have an artificially desiccated tardigrade on the wax paper, as well as the desiccated tardigrades on the moss. The way we do that is we use a stereoscopic microscope to look individually into moss, and we find the little tardigrades and we stick them to a sealed environment, and then we use potassium sulfate to create a humid environment in which the tardigrades can desiccate slowly, which is similar to their natural environment. And when the tardigrades come back from the International Space Station, we rehydrate them using water, and we look at the motility and natural functions to determine if they are truly alive. And once we do that, we look at the percentage calculations, and we are able to tell if tardigrades are truly viable in a microgravity environment.
We initially began by acquiring tardigrades from Carolina Biological Supply. And after doing so, we did several test runs to check the viability of our procedure, whether or not it would work. So we did a small-scale run in which we found tardigrades from Carolina Biological Supply, desiccated them, and then rehydrated them after a short period of time. However, after these test runs, we were finding a very high mortality rate among these tardigrades. And after a little bit of research and talking to Carolina Biological Supply, we found out that their culture of tardigrades had actually adapted out of the ability to go into that cryptobiotic state and shut down their metabolic processes. And we found the result of this to be that they no longer needed that ability as a result of their storage at Carolina Biological Supply. They're kept in small tubes with a lot of water and a lot of moss for them to eat and to live on. And as a result, they never faced any inhospitable conditions. So as years passed, that culture eventually lost that ability to go into that cryptobiotic state. And this forced us to go into the wilderness, the mountains and the foothills surrounding our community, to find these tardigrades. And we did so by picking off pieces of moss, going back to the lab, and then individually picking out the tardigrades. And we were able to get enough, but the result was we had 3 species rather than 1 since we went to the wild.
If a significant number of the tardigrades prove to be viable in the microgravity condition that we're putting them in, then maybe we can figure out what it is specifically about them, such as their DNA structure or just their general composition, that gives them the ability to undergo cryptobiotic processes. And maybe even deeper in the future, we can find a way to harness this ability and implement it into our own astronauts. So for example, we could take the DNA structure that gives the tardigrade the ability to undergo cryptobiosis, put that DNA into the DNA of our own astronauts so they can undergo their own form of cryptobiosis, which would allow them to travel deeper and farther into space, which is a giant leap for mankind, I guess you could say. But tardigrades aren't the only organism that can undergo cryptobiotic strategies. Rotifers and nematodes can, but like Garrett said, tardigrades are really unique in that they're the only organism that can actually undergo all 5, which suggests extraterrestrial origins. Any questions?
Okay, are there any questions for the Laverne team? I think Dr. Tim Livengood had one.
Dr. Tim Livengood, NCSE. So, as much as I admire the awesome toughness of tardigrades, I actually believe they're from Earth, but one of the big problems that we face in planetary exploration is preventing exporting our own life to other planets where it may mess up our ability to identify life there. Do you have any recommendations for how to kill tardigrades?
So one of the, suppositions that we had going into this was that since they're able to survive these incredibly volatile environments, they must be indestructible. But what we actually found is, ironically, they're really easy to kill or die, simply because once they're kept active, they rarely stay active for a prolonged period of time. So in the lab, if you kept them in moist moss, they would die within hours or days, which actually happened to us a couple of times. So only in this ton state or cryptobiotic state they're able to survive. So I guess if you really wanted to kill a species, just put some water on them.
Thank you.
Hi, I'm Yosef. I'm from California as well. Some of you may know me. I study philosophy, so I'm far from a scientist. But when you say that the tardigrades adapted out of the ability to go through this cryptobiotic process, from my understanding, it takes years and years for something to adapt. Is this more like a term of gene expression? So when they're, or do you think this cryptobiotic process happens only when they're in these certain environments, and if not presented in that environment, it won't go through this process?
Yeah, well, so part of the reason we think that they may have adapted out of this cryptobiotic ability is because of their short lifespan when they're active. So at Carolina Biological, they're always kept active in an aquatic environment with a lot of moss, ideal conditions, to reproduce and create a large culture, which is necessary for commercial production. However, just like any microbes or flies or small insects, if they have a relatively short lifespan, they're able to adapt quickly to their environment. So what we believe and what the biologists at Carolina Biological believe is that because of this short lifespan, they were able to quickly adapt, and it just so happened that they adapted out of the cryptobiotic ability. It wasn't really a predicted thing, and they were as surprised as we were, but it just happened, I guess.
Thank you.
All right, let's thank the Laverne, California team.